it was possible only if the first one was already bound to the DNA. So in summary, we have a complex of two different proteins that are able to recognize the promoter sequence. Okay, another one. But all those data, the conclusion was the first protein was needed to be bound first in order to recruit the second one that can recognize DNA. So is it okay for this summary, or do you want that I give you a little bit more details on that before going to the second part? So this was the summary of what we saw on Monday afternoon for you, sorry. So now we're going to go for the second part of this case study, which is now we're going to study the transcription factor by themselves. No promoter analysis anymore because we did it completely. So you see the second part, the title of the second part is the study of the transacting factor. So transacting factor means the proteins that are able to bind to the cis regulatory element that are governing the tissue specific regulation of the GH gene in the anterior part of the pituitary gland. So you remember that well, anyway, we're going to see that after all. So the very beginning of the explanation of this second part, it's very simple. It's only 2 lines, but we are going to take quite a long time in understanding those 2 lines. So they say to you that a cDNA encoding for one of the transcription factor allowing the expression of the GH gene, specifically at the level of the anterior part of the pituitary, was isolated and sequenced. And they give a name to this gene, they call it GHF. So GHF is for growth hormone, so this is the target gene, and F stands for factor. So this is the growth hormone factor that is able to activate the GH gene only in the anterior part of the pituitary. Okay, so we're going to study that. So did you receive from Ms. Hang this morning new slides? Or not?
Yes.
You have them this morning? I think it was 1 hour ago, something like that. Just to tell me, because I send the slide to Hang, but I don't know if she already sent them back to you. So just to that you have them.
I received the slide 1 or 2 hours.
Yeah, I think we receive it.
You receive it, okay, but otherwise don't worry, because if you haven't received them yet, you will receive it, I mean, in the coming hours, okay? So if you have this, so we're going to shift to this slide, because we are going to talk about how, so maybe before we start with the slides, maybe you can explain me what do you know about that. Once we did all this analysis of the promoter, so we managed to find exactly which are the elements that are necessary for the transcriptional activity. Okay, so the analysis is complete. Do you know any strategy once you know a piece of DNA that is important, that is going to be recognized by a specific protein? Do you know any strategy in order to manage to clone the cDNA that is coding for this transcription factor? Have you ever studied this kind of strategy? So the only thing that you know is that there is a protein, a transcription factor, that is able to bind specifically to a small piece of DNA in a sequence depending manner. Okay, so if you only know that, do you know any strategy that will allow you the isolation of the cDNA coding for the transcription factor? You can tell me if you don't know or if you have any ideas, and I will explain you
I
what.
personally don't know.
So you never, it's just a question. It's not, so you don't have any idea how from the simple fact of knowing that a protein is able to bind to a specific sequence, how to clone the cDNA of this protein, coding for this protein. None of
I
you,
don't.
no, okay, it's not a problem. Don't worry. You will know after. None of you know or have any idea?
I think maybe we will determine the amino acid sequence of the protein. Then we go back based on the sequence we will have
Yes,
the
you are completely
RNA
right.
sequence.
Yes, you are completely right. This is a very important step and a very complicated. So you are saying that once we have the amino acid sequence of the protein, we can use the genetic code to imagine what is the sequence, and then we can use the protein sequence in order to isolate the cDNA. That's very clear, and it's completely right. But the problem in that case, and we are going to talk about that, is how can you get the protein sequence of this transcription factor? You see what I mean? Because it means that you need to have the capacity of sequencing this protein, or this protein if it is a different complex. But in order to sequence a protein, you need to have it purified completely. Otherwise, it's completely impossible to get a sequence from a protein. Okay, but you are completely right, and we will see that it is one of the possibilities that we can have. So I'm going to shift to the slide that you received. So I stopped sharing an artificial. Okay, I go back to this one. So we are going to create an artificial transcription that is going to contain only the GAL4 activation domain, so the upper part of the GAL4 transcription factor. So it's the part of the protein that carries the ability to activate RNA pol 2. And we are going to construct a library, a cDNA library, that contains all the cDNA present in the tissue of interest that is translationally fused to the GAL4 activating domain. So we make a library, that will contain 1,000 of different clones, because all the cDNA are going to be cloned in one specific primer vector. Every clone is going to be able to express one cDNA specific to the tissue, fused translationally to the GAL4 activation domain. And what is going to happen is we are going to look at the right part of the slide, and then I'm going to make you a drawing on the whiteboard. So the strategy, I explain you first the global strategy, and then we are going to see what's happened technically for the positive clone. So the global strategy is to use a reporter like that. I'll show you the maps of the plasmid after. And we just have, for this part of the drawing, we just have to keep to focus on what is used in this construct. So technically, we've got a reporter gene. So this reporter gene can be like the Gus gene, can be LAC-Z gene, so it's a reporter gene. For yeast, there is a specific things that is totally different from bacteria. We cannot use antibiotic resistance genes for reporter gene, because yeast is a eukaryote, and eukaryotes are not sensitive to the prokaryotic antibiotics. So we cannot use, for example, ampicillin resistance or kanamycin resistance, because naturally all the yeast are resistant. Anyway, we have a reporter gene, and the reporter gene will be the use of the reporter gene is for us to be able to monitor if the transcriptional activity that is going to be conferred by the upstream sequence is going to be active or not active. Okay, that's the point. So we've got a reporter gene to test the transcriptional activity. The second part, which is the most complicated one to do, is we're going to find out the cDNA of the transcription factor we want to clone among a cDNA library. Okay, so we have first to prepare a cDNA library from the tissue of interest. Then we are going to clone this cDNA library in a specific plasmid that is going to be able to express, this is what is written here, a cDNA fusion protein fused, actually it's not written here, to the GALC4 activation domain. And then, so these are the steps. So I show you just the first step there, and then I'm going to show you a drawing to explain you how it works. So this is the first step for the cDNA library construction. So this corresponds to the completely top right part of the drawing on the right. So we start with the extraction of RNA. We then produce the first cDNA strand, then we produce the second strand of the cDNA. So at that stage, we've got double-stranded cDNA that correspond to every single messenger RNA present in the cells. And all these cDNA are going to be cloned in different plasmids. And I'm going to show you what we are going to do after the cloning and what is the composition of this plasmid. Okay? Is it okay for now? It's a little bit complex, but you will understand very easily after. And if you want, we can come back on this slide after. So I shift to the that you see. So the idea is this one. So okay, I've got the mouse here. So in red, I'm going to write down the mRNA. So we've got the mRNA. I put a poly A tail, okay? From this mRNA, we are going to make the double-stranded cDNA. So this is the cDNA library. Okay, so I write two-strand cDNA library. And this cDNA, so it means that we have a library at that point that contains all the copy of the RNA in cDNA, so we should have all the genes that are expressed in the cells. And among all these genes, you have the cDNA that correspond to the transcription factor that you want to clone. On the other hand, after I show you the plasmid, on the other hand, we've got, so it's schematic, the GAL4 transcription factor, which is completely normal. So there are two parts in this GAL4. We saw it before, an activation domain and a binding domain. The binding domain is the one that naturally recognizes DNA in the yeast in order to start transcription. What has been done, the strategy for this one hybrid yeast screening is we know in our side that we manage to isolate the piece of promoter sequence that contain the cis regulatory sequence of the gene of interest. Okay, and we know that there is a transcription factor that can bind to this sequence, and we want to clone the cDNA of this transcription factor. Is it okay for now? Yes or no? Okay. The strategy is we're going to we're going to take the cDNA, all the cDNAs, so this is why it's a library. We're going to fuse them in a plasmid in order to produce a translational fusion between the coding sequence of the cDNA to the activation domain, to the activation domain of GAL4. Why? So the strategy is like that. To produce proteins, in blue, it will be all the proteins that are coming from the translation of the cDNA in the library. And the plasmid, we will see just after, allow us to clone them just before or after. There are two strategies. We can put it either in the C-terminal region of the cDNA protein or in the NH2 terminal region. Anyway, we make a fusion of only the part which is the activation domain of GAL4 fused translationally to the sequence of every cDNA that are present in the library. So what is going to be produced? This type of protein, of construct, will produce a protein that is the fusion of our transcription factor, okay? So every cDNA that are or not transcription factor, which are fused to the activation domain. Is it okay? This part. Then, with this transcription, with this fusion, this protein fusion, we can test, and this is the idea, the capacity of this recombinant protein to be able to activate transcription in a reporter system. And what is going to be the reporter system? The reporter system, don't worry, everything is on the slide after. The reporter system will be a construct that a second plasmid that we are going to introduce in the yeast that contain, okay, so it contains a minimal promoter, so a Tata box, okay, which is going to control a resistance gene, okay, so it's a reporter gene in yeast, and I write it in red. Just before the Tata box, we are going to put from 5 to 10 repetition of the sequence, sorry. So this is the cis regulatory sequence of interest. Okay, so we can make it. Do you understand what I try to draw in this whiteboard? So basically, if you follow me, you will see it's quite easy to understand. We've got two constructs. One that contain, so this is the library, so there are thousands of different plasmids because they contain, all the plasmids contain a simple DNA. So there are effector constructs. Every effector construct is composed by a translational fusion between one cDNA and the activation domain of GAL4. So it makes a chimeric protein that contains the activation domain. In the same yeast, we have introduced another plasmid, which is a reporter system, and this reporter system is constructed like that. We've got a reporter gene, we've got a minimal promoter, and we clone just in front of the just upstream the Tata box of this reporter plasmid. We clone in multiple copies, so 5 to 10, the sequence that we manage to isolate and to identify, which is the sequence of the promoter of the gene of interest that is recognized by the transcription factor. So do you understand this? Yes or no? Okay. So if we put in the same yeast the effector plasmid, so the one that is going to produce this fusion protein, and the reporter system, what can happen? So we have two situations. So I write down once again, yes, no, I can tell you this one. So we're going to be in that situation if the plasmid, the effector plasmid, contain the cDNA that is able to, so if the cDNA of this effector plasmid is corresponding to the cDNA of the transcription factor that we are looking for. What is going to happen in the yeast? This transcription factor, which is coming from a cDNA present in the tissue of interest, is going to be translated, fused to the activation domain. The transcription factor by itself, it carries the part of the protein that is able to recognize specifically the DNA sequence that we found in the promoter. And because this transcription factor is fused to an activation domain very, very powerful, it's going, because this part of the fusion protein is going to bind to DNA, and the second part of the fusion protein is going to activate transcription. So we will have expression of the reporter gene. Is it okay? So in that case, it means that the cDNA that is contained in the plasmid from which this translational fusion is produced correspond to a cDNA of a protein that can bind specifically to this promoter sequence. If we have a cDNA clone that is not able to bind to DNA, what is going to happen? So I redraw the same. So this is the reporter, this is the data box. Here we've got the several regulatory elements that we put in front. But if we produced one fusion protein always with activation domain and another cDNA, and if the protein translated from this cDNA is not able to recognize the cis regulatory sequence that we clone in front of this reporter construct, since there won't be any interaction, the activation domain won't be able to activate transcription, and there won't be any expression of the reporter gene. So this is not working at all. Do you understand the strategy? Okay, everybody understands or not? So one question on this, according to you, why do we need to make a fusion of protein? So it means, why do we need to fuse the protein coming from all the cDNA to an activation domain? What would you say for that? Do you have an idea?
Can you repeat the question?
Okay, so you understand the concept. The concept, which is conceptually it's simple, is we are producing all the effector plasmid produce a fusion protein, which is the part of the protein that is coming from the translation of the cDNA that is translationally fused to the activation domain. So it's a single protein, and we add an activation domain in order to activate the transcription and the translation of the reporter gene. But my question is it necessary to have this activation domain fused to the protein? So I explain you why this okay. If we take back, it's important that we discuss about that. So what I'm going to draw now is for example the GH gene, the one that we study. Okay, so we managed to find. So I make a very simpler version. We managed to find that there was a cis element that is recognized by a protein, and this protein was responsible for the activation of the transcription. Okay, this is the starting point. So it means that this protein, this transcription factor, has two functions. The first capacity is to recognize specifically the piece of DNA contained in the promoter of the GH gene. Okay, and the second concept of the protein is it contains a part which is an activation domain because it is able to activate. Okay, so in theory, we shouldn't have to fuse another activating domain to this protein in the yeast 1 hybrid system. This yeast 1 hybrid system, look, the transcription factor is here, and we add another activation domain to be sure that the transcription is going to start. Okay? So if we look a little bit this strategy, we shouldn't have to add an activation domain because the transcription factor, this one, since it is an activator by itself, it contains already an activation domain. You understand? Yes or no? But we add an activation domain coming from the GAL4 from yeast, because we don't want to be very stringent in the cloning. I explain you what I mean. Imagine that we want to clone the cDNA of a transcription factor, so that is able to recognize the piece of DNA in the promoter, but that naturally doesn't carry any activation domain. For example, it's a transcription factor, but that is a repressor. For example, it's not an activator. So if we don't fuse to the activation domain, all the proteins that are able to bind to recognize specifically this sequence, we don't know if it's going to be able to activate transcription. So this is why for all the cDNA, we fuse them to an activation domain in order to be sure that if there is interaction, we are going to be sure that the transcription activity will happen. Okay? You understand or what? It means that with this strategy, we are able to clone every kind of protein that can be activator, repressor, or nothing, okay, only on the basis of their capacity to recognize specific sequence contained in the reporter gene. Okay, you understand the concept? Okay, so I stop sharing this whiteboard. I go back to the slides so we can understand. So, okay, now it's okay. Sorry, because I want to put it full screen, but so you understand now a little bit after I'm going to show you the plasmid. So we go back for that. So we generate a library. So all the different cDNA contained in the cells, they are constructed in this type of plasmid. The map will be shown just after. From that, we generate all these library protein, which are fusion protein. What is called DNA, bait DNA, it is the cis regulatory element that we found in the promoter of the gene of interest. Okay, so these are the plasmids. So we have two different plasmids. So just to show you, so there are plenty of different plasmids that have exactly the same function. Most of them, they are commercial. Okay, well, anyway, we've got, just to show you how it is, we've got the effector plasmid and we've got the reporter plasmid. So we're going to start with the reporter plasmid, the one which is on the right. So do you understand the map? What do we have in this plasmid? So this one is more precise. We have the black arrow here is a data box, only a data box, okay? The OpenReady-Frame just downstream the data box, which is called HIS3. It's a gene that is going, when it is expressed, that is going to be, it's a gene that I will explain you how we make the selection after. It's a gene that is able to, that give the possibility to the yeast to produce itself the histidine amino acid. And yeast need to have histidine in order to grow. I'll explain you how it works. So we have the data box only. This is the reporter gene, HIS3. And in front of the data box, just where you see this little black arrow, we fused, so in that case, it's only 2 copies, but the purple boxes are the sequence of the promoter, so the cis regulatory element, for which we want to find which are the transcription factor able to recognize this part of DNA. So this is the reporter. Pieces of promoter, data box, minimal data box, and reporter gene. Okay. Here you have 2 other genes, and I will come back on that. There is a gene which is for kanamycin resistance, and another gene that is able to participate to tryptophan, which is an amino acid in yeast. Okay. I'll come back on those 2 genes after, and I explain you on those ones. Okay. Did you understand the map of this reporter plasmid? So we have the pieces of the promoter, the minimal data box, and the reporter. In that case, it's HIS3. Okay. Is it fine or not? Okay. The reporter, the effector plasmid. The effector plasmid are the ones that are going to contain every single cDNA that have been produced from the tissue of interest. So the map is a little bit more detailed. So we are going just to see a little bit. We have here, so we start with the black big arrow where it's written PADH1. So PADH1, it's a promoter of yeast. It's a constitutive promoter in yeast. ADH is the gene in yeast that is alcohol dehydrogenase. So it's a constitutive promoter in yeast. This is the only thing that we need to keep in mind. So we have a constitutive promoter. Just after this constitutive promoter, we've got restriction sites. So it's HIND3 here, for example. And then we've got this small protein that can be produced. We have here SV40 NLS fused to GAL4 activation domain. So we are not going to I can tell you what it is. NLS is nuclear localization signal. It's a very small peptide that is able to target the recombinant protein directly to the nucleus. Okay, so it's just to be sure that the protein that will be produced will be localized in the nucleus of the yeast. So I come back. We've got a constitutive promoter, a cloning site, and an activation domain. Then here also you'll also have another multi-cloning site, and so on and so on. So what can be done with this plasmid is either here at that position in 3, or after at the position of the multiple cloning sites. So before or after the GAL4 activation domain, we're going to clone all the cDNA library. So when I say we're going to clone all the cDNA library, it means that we are going to produce as many plasmid as there are cDNA. So every single plasmid contains one single cDNA. Okay. And what is going to, okay, so we've got GAL4, and then what do we have here? Okay, things are missing. We have on the plasmid, on the reporter, it was the gene for kanamycin resistance. Here it's for ampicillin resistance. On the reporter plasmid, we had a gene to synthesize tryptophan. And on the effector, we have a gene responsible for synthesis of leucine, the other amino acid. Okay. I want just to show you before I explain you how it works and how technically we work with yeast. Look at this effector plasmid, and do you know what is PUC, P-U-C, ORI? Do you know? And 2-mu ORI. You never heard about that?
No.
Okay. No, it's not a problem. Well, you know what is a plasmid? A plasmid is a circular piece of DNA, but the plasmid, they are only in bacteria, mainly. I mean, almost only. So in order to replicate in bacteria, you need to have on every plasmid an origin of replication. Okay, so this point, you know it. So PUC ORI, it's a replication origin for prokaryotes, so for bacteria. There is the same here. So why we have a bacterial replication origin is because all the molecular biology techniques, the cloning, the cDNA synthesis, the ligation, the transformation, it is done in Escherichia coli, in E. coli. We don't never work with yeast to make construct. It's too much complicated and almost impossible. So you have to keep in mind that all the molecular biology steps, which are plasmid construction, ligation, and so on, everything that is cloning is done in bacteria, in E. coli. So those plasmids, they are construct in E. coli. And this is why, in order to be able to have multiplication in E. coli, we have the origin of replication of E. coli present in both of the plasmid. So it means that the construct, the cloning of the reporter plasmid and the cloning of the library in the effector plasmid, everything is done in E. coli. So all the manipulation, all the cloning experiment. Then, once all the constructs are done in E. coli, so the reporter construct in one side and the library on the other side, all those plasmids from E. coli are extracted and then transformed in the yeast. So a plasmid can live in the yeast only if it contains a yeast-specific replication origin. And the yeast-specific origin of replication is called 2-mu. So a plasmid that contains a PUC origin and a 2-mu origin means that it can replicate and it can stay inside E. coli and yeast in two different systems. You understand? So it's a plasmid that can be used in E. coli and then can be used in yeast. So technically, what I was saying is all the construction cloning are done in E. coli and the analysis is done in yeast. Okay? So I want to come back on this. So I told you before, so just to go on a little bit on that, because there are plenty of things to explain you on this plasmid. So each of the plasmid contain an origin of replication specific to E. coli. Perfect. So those plasmids can be multiplied in E. coli. They contain, both of them, resistance to antibiotic that can be expressed only in E. coli. For example, the PGAD-T7AD plasmid is in E. coli is ampicillin resistance. We'll give the ampicillin resistance. For the reporter plasmid, the PHIST2.1, it is a plasmid that will confer kanamycin resistance to E. coli. So technically, we've got origin of replication for E. coli and two different resistance for antibiotics in E. coli. Is it okay? This. They are different. It could have been ampicillin here as well. It's not a problem. Anyway, this is prokaryotic antibiotic resistance. Is it fine?
Yes.
Okay, but as I told you, antibiotics doesn't work in eukaryote. So it means that this plasmid, when we are going to transfer them in the yeast to make the screening of the library, we cannot select them because of their capacity to confer antibiotic resistance. So what we use instead of antibiotic resistance that is used in E. coli, what we use in yeast is called something. So you have to tell me if you have ever heard about this name. It's called auxotrophy. Have you ever heard about that?
No.
It is extremely simple. Don't worry. So you all know antibiotic resistance for E. coli.
Yeah.
Okay. So you select the bacteria that contain the plasmid because the plasmid brings the antibiotic resistance. So I shift back on the whiteboard to explain you what is auxotrophy. And it's very simple, you will see. And this is the equivalent, it's not at all, why it doesn't work? Okay. The equivalent of antibiotic resistance. So, in yeast. So, yeast are able to synthesize by themselves plenty of different amino acids. So a yeast can synthesize leucine, tryptophan, alanine, histidine, all the amino acids. It is completely autonomous. Okay, no problem. But we generate genetically modified yeast with some mutation in some of the genes that are implicated in the biosynthesis of this amino acid. For example, we can have, a yeast, and the genotype of this yeast will be is minus. What does it mean? This yeast, it's a totally wild type yeast, except that one gene, which is implicated in the biosynthesis of histidine, the amino acid histidine, is mutated. So it means that this yeast cannot grow if you don't put in the growth medium, if you don't put histidine. Okay? So you will have, if you put this yeast in a medium where you add the amino acid histidine, there will be a growth of the yeast. You understand? It's very simple. The yeast has been genetically modified in order to knock out one of the genes that is implicated in the biosynthesis of the amino acid histidine. So this yeast cannot grow by itself unless you give the yeast directly the amino acid. And in that case, it can grow. It can live. So if you take a yeast that its genotype is His minus, and you put a plasmid like the one we saw that contain the gene Is plus, you will complement the mutation, and it means the yeast is going to grow. Is it okay? Yes or no? Do you understand this or not?
Yes.
Okay.
Yes.
Okay. So this is what we call it's. You see, it's very similar to antibiotic resistance, but in the other way around. Okay. So we don't call that antibiotic resistance because it's not antibiotic resistance. It's called auxotrophy. So it's written like that. Auxo, T-R-O-P-H, auxotrophy. Okay. So we say that, for example, this yeast is auxotrophic to histidine because you need to have this yeast to grow. We need to either grow them in a medium that contain histidine, or introduce a plasmid in this yeast that is going to complement the mutation. Okay? So from this, it's quite easy to understand the following step. So we are in a situation that in one yeast, there will be all the in the plasmid, so this is the reporter plasmid. We put the cis element sequence in this plasmid, and this plasmid is able, if I remember well, but it's not a problem, to complement yeast that are mutated on the leucine gene. I explain you how it works after. I need to remove this. So, okay, it's like that. Then, on the other side, we've got the fusion of the protein with the library that in some cases are able to bind to the DNA and to activate transcription. Okay? So, if we put, so this is the effector. So if we introduce the effector plasmid and the reporter plasmid in one single cell of yeast, if the effector construct is able to recognize specifically the element that we clone in the reporter plasmid, this interaction will give rise to activation of the leucine gene, and if the leucine, the yeast has a genotype that in which it cannot synthesize the amino acid leucine by itself, if the fusion between the activation domain and the cDNA binds to the promoter, the transcription of the leucine gene from the plasmid will be active and will complement the mutation of the leucine in the yeast. So this means that this is going to be produced and you will have expression and complementation of the mutation. And so it means that the yeast, they will grow. Do you understand the principle? Yes or no? So this is what we call auxotrophy. In other words, it's quite simple. You take a yeast that does not contain one function. In that case, the one gene in the biosynthesis of one amino acid. And if our construction works, in other words, it is able to activate, to bind and then to activate this gene, then it will complement a yeast that is mutated in this gene. So it means that when we are going to screen our library, because we have to keep in mind that all we want to do at the end is to clone the cDNA of the transcription factor. So we put all the cDNA there. This is the reporter construct. And you see, so in the drawing that I did, I wrote leucine, but in that case, it's histidine. So it means that if I have one yeast that contains one plasmid like that with the cDNA of the transcription cloned here or here, it means that it's going to bind to the regulatory sequence in the reporter plasmid. From this binding, the histidine-3 gene will be transcribed and translated, and therefore, in the yeast that contain both of the plasmid, they will be able to grow on a medium that does not contain histidine. So it means that the screening will be finding among all the yeast in the library, the one that are going to grow on a medium that does not contain histidine. And those yeast that are able to grow in such a medium will contain obviously one cDNA that, when fused to GAL4, is able to activate this gene. So it means that the cDNA clone inside this plasmid is going to code for a protein that is able to bind specifically to this promoter region. Is it clear? Or not? So the strategy, we can say it is summarized there. So we got two partners, okay? The DNA bait, it's the piece of the DNA of the promoter for which we're looking for the protein that is going to bind there. And the prey is the transcription factor. So look at the strategy. This is exactly what we saw before. We generate a CD library fusing activation domain to a putative transcription factor. Okay, we have the origin of replication for yeast, no problem. And then for the bait construct, which is the reporter construct, we put the piece of promoter, this is the yellow box, or the bait DNA, and the reporter gene. So you see auxotrophy, and so on and so on. Once we put the two plasmid in the yeast, if there is this situation, this configuration, the reporter gene will be active, and only the yeast that are going to grow will contain this configuration. And from that, the only thing that needs to be done is to extract back this plasmid and to sequence the plasmid in order to have the cDNA of the library, the cDNA of the transcription factor. Is it okay? Or not? Okay, so just but anyway, it took a little bit of time, but it's not a problem. So you see this strategy, it's very interesting because everything is done in vivo, in yeast. Yeasts are very easy to manipulate. So there are one pro feature of this one hybrid system. It's a very fast method because you can almost do a full library screening. So starting from the construction of the library, including all the cloning steps and the screening, you can say that you can do a one hybrid screening in less than 2 months. So that's not a real problem. So using this, you can easily clone the cDNA coding for a protein that is able to recognize specifically the piece of DNA that you already isolate. There is a very bad drawback anyway. This strategy generates a lot of false positive. So it's very complicated to explain. I did a few years ago quite a lot of one hybrid screens, and it is very powerful. It works, but you generate a lot of false positive. So where they are coming from, this false positive, is very complicated to understand. But anyway, it works. But there is anyway a very important technical limitation of this strategy. It is what I wrote on the last point on this slide, and I would like you to discuss with me about this point. This strategy, the yeast one hybrid system screening, works only for monomeric or homomultimeric transcription factor. And I would like that you try to tell me why it works only in situation. Do you have an idea? I think it's better that we discuss about that because it's the best way that you can understand the process, I think. So do you understand the question? The synthesis, it's a fast method, no problem. Fast, quite easy, cheap. Cons generate a lot of positive, but it is not, both of these points are not important. It's fast, okay. you have a lot of false positive. Okay, it's not a problem. You sort them after. But the real problem is that it only works for monomeric or homo-multimeric transcription factors. So do you have an idea of what I want to mean when I write this? We are here to discuss it, don't worry. So tell me what you think about that. It's not with the O, it's fancy. You have an idea? Xiang, tell us what you think.
I think it is hard, almost impossible to try with like heterodimer, for example.
Why? You're right. Heterodimer, it's impossible. Why?
Because I think like with the monomeric, we already try with all the cDNA in the organism. If we want to find the heterodimer, we have to try every combination.
You're right.
There is.
Yes, but is it possible to try combination? That's another point. Is it technically possible? I understood that you understood. Technically, is it possible or yes? Yes or no?
I would argue that it is not impossible, but it is so labor.
Okay. So you would argue that it is not technically impossible, but it will take so many time to test all combination that it's not feasible. Is it right?
Yeah. Yes, I think so. Off the top of my head, that is.
Yeah, I understand what you mean. So you are partially right and partially wrong because, okay, so I explain you. Okay. So I will make a drawing very simple. Imagine that the situation of our gene, okay? So we know the cis sequence, okay? No problem. And we know that the transcription factor, okay, it is an heterodimer. Okay. And once it is an heterodimer, it works. Okay, no problem. So if we want to isolate the cDNA from such a situation, it's impossible. Wait, one point. It is the only situation, so you need to have an heterodimer to have it functional, okay? Every single monomeric unit doesn't work alone, okay? The problem that we have is when we make a cDNA library for one hybrid system, okay? Every protein is encoded by one single cDNA. So that's the basics. And in the yeast, you transform the yeast with one single construct, effector construct. So it means that all the yeast, I will show you two situations. First yeast and second yeast. So imagine that you have thousands and thousands of yeast. All those yeast, they contain the reporter construct. Okay, so this is the reporter construct that contain the piece of DNA of the promoter. Do you agree with this? So this is the reporter construct in one plasmid. But every yeast contains one single plasmid containing one single cDNA of one single effector. So it means that the first yeast, for example, can contain the cDNA of the red protein fused to the activation domain of GAL4, But the other yeast contain the other monomer of the dimer fused to activation domain. Do you follow me like that? So it means that the first yeast, the one on the left, is able only to translate the red monomer, and the yeast on the right is able to translate only the blue monomer. So it means that in those two different yeast cells, you cannot be able to produce both of the monomers in order to create the dimer. And it is if the heterodimer is the only active form, there is no possibility of finding a yeast that contains two different cDNA at the same time. So you understand the problem?
Yes. So we cannot create an effector plasmid containing more than one cDNA. Okay.
No. Every effector plasmid contains only one cDNA. This is the proper definition of cDNA library. You've got only one cDNA clone in one plasmid, one vector or whatever. So it is complicated because you see, technically it should work, but the problem is that the two different monomers that form the heterodimer are physically in two different yeasts. So they will never be able to interact together, and it won't be completely impossible to recreate the heterodimer, which is the functional form, in fact, of the heterodimer. You understand? So that's a problem. That's a problem because there are plenty of situations in which. So, for example, if we go back to the exercise we did on Monday morning. So you remember when we studied the promoter, there were two sites of interaction. One that was autonomous. So there was one transcription factor that can bind directly to this site, and the other one needed the presence of the first one. If both of these complexes are only a single protein, only the first protein, transcription factor, can be cloned with this strategy. You understand? Because the other one, the one that needs the presence of the previous one first in order to bind, you will never find it using one hybrid system. Okay? But it's not because of this big problem that it is not used at all. It's very useful because it's very fast and you can find very good candidates. Once you've got one starting point, you've got one candidate, you can still go on trying to find other interactants. But the biggest problem in this strategy is that situation, the one that I wrote here, is when we have the transcriptional activity is carried out by an heterodimer, and this situation cannot be solved using yeast one hybrid system. It's not possible. Okay. So I propose you to make a break now because we have finished. Before having a break, do you have any question about this one hybrid system strategy, or is it okay?
It's okay.
It's okay. So I propose you, because I have a few phone calls to give, to make a 15-minute break so we can start back at 35 if it's okay for you. And what we are going to see is the other strategy, the biochemical approach, the biochemistry approach, so which is the purification of transcription factor. And you will see that in that case, we can purify complexes, but we will spend less time than on the yeast one hybrid system. Okay, so we join back in 15 minutes. It's okay for you? Now we're going to shift to the second strategy. So it's the following slides. So the second strategy, as I was showing you at the very beginning, it is the biochemical approach. which basically concern the transcription factor purification. So we will be really quicker than the yeast one hybrid system because it's technically it's more simple, really more simple, but experimentally it can be quite difficult. So the idea is to try to use the classical biochemistry purification strategy in order to purify the transcription factor. And as somebody was saying just at the beginning of the lecture, the idea behind that is once the transcription factor or the complex, and this is the big difference between the one hybrid system, once the protein, so the transcription factor complex is purified, we can micro-sequence the protein and then have an idea about the sequence of the cDNA and try to find directly the cDNA. But the most difficult part is to purify the transcription factor. So there is, in the slide, I only put one slide, I will put it full screen. Yes, one slide to explain you the global strategy, because as I was explaining you, it will depend, every situation, every specific case will be a little bit different. But nevertheless, there is a common global strategy for purifying protein that have the capacity to recognize the specific sequence of DNA. So the strategy, I explain you very quickly using this slide, and then I will try to draw some specific point on this strategy. So the idea is to do a two-step, because there are two conceptual steps in this type of purification. So if you look at carefully what is written on this slide. So it's a slide that I've taken from a book. So it's maybe a simplified version of the strategy. So the starting point is on the left of the slide. So step one. First of all, you will see it's not the case when you are in the lab doing the process. It's not, we don't start with total cell protein. So basically we start with a protein extract. Okay, and you see in the drawings on the slide, we use classical chromatographic columns, but we matrices that are specifically dedicated to the purification of protein that have an affinity to DNA and sequence specific. So the idea is the first step, the step number one, is from all protein contains either in cell or in nuclei to sort the protein based on one particular biochemical feature, which is their capacity to recognize DNA. So you see here, they show you that you put the mix of proteins, so the total extract, you put it on the first column, so the first step, and it's written the column with a matrix containing DNA of many different sequences. Okay, actually we don't use that now. Now it's a little bit a wrong way to explain the things. So the idea of this first step purification is to separate from a mix of total protein two kinds of protein: the protein that have a general affinity to DNA and the protein that does not have affinity to DNA. Okay, so we want to separate the protein that can recognize double-stranded nucleic acid and the protein that cannot recognize double-stranded nucleic acid. So this is the first step. And if you look, so we check the two steps first, and then I will explain you more in detail the different steps. So first step, we separate the protein with DNA affinity from the protein that have no affinity with DNA. So at the end, we end up with two different extracts. The one for which we don't have any binding activity, and a fraction of all the protein that have a natural affinity to DNA. Okay. Among all those proteins, you have protein that can bind specifically to the sequence of your promoter. So in that example, the sequence we are looking for is GGGCCC. So we are going to use the step number 2 in order to purify among all the proteins that have a general DNA affinity, which one can specifically recognize this sequence and no other DNA specific sequence. So we are going to use another type of colon, which is a colon that you have to make yourself, because it's not commercial, because you will understand that this colon, they contain a matrix, like all the colon in biochemistry, but this matrix is linked covalently to the DNA sequence of the cis regulatory element of your gene of interest. And using this second column, you can sort from the fraction that contain all the proteins that have a general DNA affinity. You can sort the one that can bind specifically to your sequence from the other one that does not specifically recognize this type of sequence. And at the end of this second step, you end up with two different fractions, proteins that are not able to recognize and to bound to the sequence, so we don't need this one, and the proteins that are able to bind to this sequence that you covalently linked to the matrix of the colon. And it is this fraction, so the last fraction, that is going to be sent to specific services for protein sequencing, and so you can have the protein sequence from that. Do you understand this two-step strategy? The first one is general purification of proteins that have affinity, general affinity to DNA. And then second step, among these proteins, we sort the one that have an affinity to your sequence from the one that does not have any affinity to your sequence. So I just come back on the first step. In this slide, it's written, okay, that the first column is a column with a matrix that contains many DNA sequences of different sequences. This is to mimic DNA without any sequence requirement. Actually, what is done, instead of putting in the first step column plenty of DNA fragments with different sequences, we use something that is very different. I don't know if you ever heard about that. So the little ball is here to represent the matrix of the colon, okay? And instead of covalently linking a piece of DNA with all the sequence, the potential sequence, DNA sequence, we don't put DNA, actually. We put something which is a polysaccharide that is called heparin. Have you ever heard about this heparin?
No.
It's not a problem. Eh? Okay, basically the strategy, conceptually, it's exactly the same as this. This polysaccharide, if you look at how it is made, okay, so it's a polysaccharide, for the protein that have a general affinity to DNA, it mimics DNA completely without any sequence requirement. It means that for a protein that can recognize DNA, it can recognize heparin without sequence requirement. So, for example, this first sugar can be A, T, G, or C. It's the same for the protein. So it's a kind of general DNA, like a piece of DNA without any sequence. Do you understand? It's quite difficult to explain this kind of things. So it's a linear molecule, okay? It's a polysaccharide molecule, but the behavior of this molecule, it behaves in this situation exactly as if it was DNA, but without any specific sequence. So it's like general DNA. You understand? And so I will shift to the whiteboard. Just 1 second to explain you here and here. Okay. Does this work? Yes. So if we want to to make just a quick, we're going to spend, let's say, 15 minutes maximum on that. Okay, so this is double-stranded DNA. Okay, general DNA. We have two kinds of protein that have an affinity with DNA. We've got the proper transcription factor. Okay, so what we know for this transcription factor is they can recognize specifically a specific sequence of DNA. So I would take the same example, CCC, GGG, for example. It can recognize only this. Okay, so this is the case for most of the transcription factor. After, do you know protein that can recognize DNA, but without the need of a specific sequence? Like protein that can interact with DNA, but they are not interacting with DNA because of a specific sequence. They just recognize DNA in general. We talk about some of them at the very beginning of the course. Which one, Xiang? Do you have an idea?
I do remember the concept, but right now I can't think of any specific.
No, but in terms of protein that we saw, for example, a transcription factor can really recognize a specific sequence in a promoter, and it can recognize only the sequence. Like a restriction enzyme, for example, if you take EcoR1, it recognizes GGA TTC, final point. It recognizes only this. So there is a sequence-specific necessity for the interaction. But you have in the nucleus plenty of protein that just recognize DNA, but not a sequence, not a specific sequence. This is the case, for example, for the histones. The histones, they recognize DNA when they are in the octomeric form. They just recognize DNA, but they don't bind to specific sequence. Okay? So among all the proteins that can interact with DNA, actually, there are two families of protein. The one that recognize specific sequence and the one that recognize only DNA in general. Okay? Do you follow me? Is it okay?
Yes.
Okay. So the global strategy, and this is a general strategy for purification of transcription factor. The first step in every purification is whatever it is, a transcription factor, an enzyme or whatever, you need to, because you will increase the efficiency and the yield as well, is you need to purify the organite in which you suppose the protein to be. Okay? So in this lecture, we are talking about transcription factor, where in which organite you are going to find the transcription factor. Very simple. It's in the nucleus. You agree? So the first step always is to purification of nucleus. Okay, first step. Second step, you make a protein extract of the nucleus. So at that step, we have two kinds of protein. The protein that have sequence, that can recognize sequence specific region, specific, or sequence non-specific. This is what we talk about, specific. Okay? Okay, from that, the third step, we're going to make this general DNA affinity column. And this is the column that contains heparin. So from that, we are going to use a column like that. So I'll just move a little bit up. So we use a column that contains heparin. We're going to put on the top of the column all the protein that has been extracted from the nucleus. So what is going to happen here? We are going to keep only, so we are going to sort out exactly as the same style. Only we're going to collect one fraction with all the protein that can interact with DNA. And in this mix of protein, you've got the protein that are able to recognize general DNA or sequence-specific DNA. And it is this type of extract that we are going to use in a colon. So I make you, so the colon, it's like beds of things. And what we do is covalently, we link the sequence, for example, GGGCCC. We link the DNA sequence to the matrix of the colon. And this extract that is coming from the first heparin Sepharose matrix, we're going to load that on the second one. And in that case, only the protein that are contained in this extract that are able to recognize this sequence will be linked to this, to the matrix. So all the one that does not have interaction with this sequence are going to be removed from the colon. And at the end, we're going to make the elution of only the protein that are bound to these pieces of DNA. And from that, we will manage to get only the proteins that are in interaction with this specific sequence. So do you understand the strategy? It's okay? So the advantage, so I make a kind of big zoom on the last chromatography. So this is the matrix. Let's imagine that here you've got your sequence, G, 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 C, so C, 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 and so on, okay? What is very interesting, so we use, sorry, I didn't tell you, we use double-stranded in order to be really in the same situation as DNA. So what can happen there? And this is where this strategy is different from the one hybrid system. So in this context, we're going to load on the colon all the protein, but exactly in the same context as they are inside the nucleus, because we took a lot of care in preparing the extract. So for example, if we are in a situation where the transcription factor that is able to recognize this sequence, it's an heterodimer, okay, let's say like that, for example. But you need to have, as I was saying before, both of the monomer, the blue one and the red one, to be able to interact with this sequence. So this is the case that wasn't possible to be used for one hybrid system, because we couldn't have both of the protein in the same yeast cell for the yeast one hybrid system. But in that case, because we do just biochemistry, so we extract the protein and we keep the interaction between all the protein all together as they are in the cell. So it means that in that situation, the heterodimeric situation is conserved. We didn't separate the red from the blue, in other words. So it means that in that situation, we can have all this complex that is able to be bound to the target piece of DNA that is covalently linked to the matrix of the colon or chromatographic colon. So you understand that in this way we can purify heteromeric complexes that we won't be able to study using one hybrid system. You understand that? Is it okay? And on the top of that, there is even more interest in that, because I erase this. I just remake the drawing here. So imagine that we were in the context of this is, as I was showing before, an heterodimer that is able to bind, to recognize specifically this sequence, okay? So using this strategy, we can easily clone it, okay? Purified it, I would say. But imagine the situation where the complex is much more complicated. So there are only two monomers when in interaction together can recognize the DNA. Okay, so this is the binding domain of the complex, the domain of the complex that is recognizing the DNA. But you saw that in plenty of examples that there are so many proteins in interaction, so we can have another one in the complex, let's say a green one, or another one. Okay, so let's imagine that it's a big tetrameric complex. But in that situation, if you look this completely artificial example, we have an heterotetrameric complex in which only two of the four partners are able to recognize the DNA, and two other, the green and the black one, they are never in contact with DNA, but they are part of the complex. Okay, so this is the general complex that is bound to the DNA, but only two of the monomeric units recognize DNA. But using this biochemical approach, which is basically purification based on DNA affinity, you will manage to purify the whole complex. And that's very interesting because at the same time, you are going to purify the partner that are in direct interaction with your target, and at the same time, other proteic partners that were in interaction with the one that are directly recognizing the DNA. You see what I mean? So it means that with this strategy, you are able to purify the complex, and this complex, you are able also to purify part of the complex that are never in direct interaction with DNA, like the green and the blue one. Okay, and from that, once you manage to purify this complex, the following step are pure biochemical analysis. So it means, what can you do with this? You can use SDS page gel separation of protein to see using beta-mercaptoethanol and so on to see if there is disulfide bonds and so on and so on. I mean, you can do a proper biochemical analysis of the complex. You can count the number of subunits. You can sequence the subunit. So when you sequence the subunit, as it was said before, you can then get the cDNA. So you see the difference between I go back to the slides. So this strategy is very powerful. It's quite long. The advantage of the purification, of this type of purification, is that, as we just said, we can purify heteromultimeric transcription factor complexes. That's very, very important. And you have to keep in mind that it's maybe the most common form of transcription factor. They are very, very often engaged in multimeric complexes. The cons of this type of strategy, it can be very time consuming, okay, because you need to do plenty of steps of purifications. Sometimes you need to do optimization of the condition of binding. So this is biochemistry. It's very more complicated in terms of setting up an experiment. And what can be complex, but nowadays it's easier and easier, is you don't have to forget that at the end, what you have purified, you need to sequence it in order to identify the cDNA of the transcription factor. So you see that from this summary of the pros and the cons, this approach is completely complementary to the one that we saw before, which is the yeast-1 hybrid system. The pros are different and the cons are different in both of the strategy. So if you want to make a kind of summary of these two strategies, I would say that you will have, if you are working later on in this kind of transcription factor identification, you cannot choose before which is the right strategy that is going to be fruitful. You need to know a lot of things about the complex formation in the promoter that you are studying. You need to know if it is multimeric, monomeric, and so on. And usually the researcher, when they want to isolate a cDNA coding for a transcription factor, what can be done first is to try one yeast, one hybrid system, because it's quick. It can be very efficient, but if it doesn't work, it's not a problem. And usually the researcher, they start after yeast one hybrid system, they start transcription factor purification after. So it's very rare to see a researcher directly starting with the purification of transcription factor because it can be quite tricky. Okay. Do you have any question on this part? So these are basically the two strategies that you can use to clone the cDNA for transcription factor, either yeast one hybrid system or purification. Do you have any question on this part?
No.
Or is it okay?
It's okay.
So you understood that it's not, they are not the same strategy, they are complementary. Okay. After, we will going on the exercise, don't worry. I have to stay with the slides because since we are talking about this interaction between transcription factor and cis regulatory sequences, I prepared you a few slides, which is the reverse analysis. So I go back to the whiteboard. I'm sorry for changing all the time, but usually when I'm doing this lecture, I have a real blackboard and I'm using it all the time. So using the Zoom, it's not convenient. So it's just to show you the link of what we are talking about. So from now, we did this, we did all the analysis in this way. We know we identify the cis element in the promoter, and what we just saw now is we can clone the transcription factor. Okay, so in other words, we started from the regulatory sequence contained in the promoter, and we used this regulatory sequence in order to clone the cDNA or to purify the complex, to clone the cDNA of the transcription factor that can recognize this cis element. So this is very, very common strategy. There are also the other strategy that you need to know is sometimes you are studying a transcription factor because it's one of your target gene, okay? So your interest is one specific transcription factor that has been characterized in one situation. And the question that you ask yourself is, what are the target gene? You understand? The question is, you have one transcription factor, which genes this transcription factor can recognize? And it's the first step in order to make network of gene regulation, because you can have one transcription factor that is able independently to activate gene 1, gene 2, gene 3. And what you want to know, you know the transcription factor, it's okay, but what are the genes that are going to be regulated by this transcription factor? Okay. So I prepared a few slides just to show you the strategy. And it's a general strategy, so I cannot give you more detail because it's very general. So the idea, you know the transcription factor, and the question is which gene is going to recognize. So we are going to look for DNA binding. So we have, so you all remember of the method of electromobility shift assay. Is the one that we used in order to understand what was the interaction between the proteins and the DNA. So this little, so if you see the mouse, I'm going to put the red. If you look this electroradiography, it's a typical gel shift assay, EMSA. So you remember we put the probe, the piece of DNA of the promoter, we incubate it with the proteins, and if there were an interaction, you have a mobility shift, and this is the complex formation that can be seen on the gel. Okay, so the idea of this strategy, which is called binding site selection, is to use, and you see it's not complicated because it's exactly the same method. It's electromobility shift assay, but used in the reverse way. So what is done is, you know your transcription factor, okay? Because it is your starting point. You have a transcription factor and you want to find which are the binding site that this transcription factor can recognize. So you are going to prepare an artificial piece of double-stranded DNA that is very simple. And we are going to use this double-stranded DNA as a probe in the electromobility shift assay. So this piece of DNA is composed by two blue regions. So these are the sequence of the blue region. You can put whatever sequence you want. The only thing that you need to know is what is this sequence, because we are going to use primers that are complementary to this sequence. So technically, we have one piece of DNA in which the sequence on the right and on the left, they are none. And in the middle of the sequence, that is represented by the orange box, we put a random double-stranded DNA sequence. And I wrote N all along, which means that we have 20 base pair of random DNA sequence. So for example, the first position can be A, T, G, or C, second position the same, and so on and so on. And we are going to do electromobility shift assay using this DNA fragment as a probe. So the DNA fragment, okay, sorry, somebody is entering the room. It doesn't work. So I've got a problem. Excuse me, I've got a small problem. Okay. Because one student is coming in the room, I don't know. So we're going to use, sorry, I start again. This fragment with a random sequence in the middle, we're going to use it as a probe for electromobility shift assay experiment. So the random probe is drawn here with the little black box. The little black box is the random sequence. And the way I draw this drawing is, in the middle, this random sequence can be any sequence possible, completely in a random fashion. So I just try to highlight three different types of sequence. So there is the orange one, which is a specific sequence, a red one, which is another one, and the blue one is the sequence that has been put here that is recognized by the protein that is giving rise to your complex. So if you start with this as a probe, it means that the probe is composed by a mix of different sequences. So you can load the probe in one lane, okay, same size, but different sequences. Then you put the same mix of sequence that are the probe, you incubate it with the protein. You will have some sequence of the probe that will be able to interact with the protein, but others, because this is not the right sequence, won't be able to interact with the protein. But nevertheless, what you are seeing in the gel shift experiment, so the band that have a reduced mobility, this band corresponds to a situation in which the transcription factor that you are studying has recognized a piece of DNA because it's only the DNA that is radioactively labeled. Okay, so what is done? The concept is this one. You put a mix of probe, you add the protein that you are working on, you collect only the protein that has linked to the piece of DNA in interaction, so the complex, and you collect the DNA that is inside this band. From this, you can do several rounds of successive rounds of purification. And do you understand that at the end, the DNA that is contained inside this complex will contain only the sequence that is recognized by your transcription factor? Do you understand this concept? Yes or no? Or what do you understand if you don't understand?
Yes, I understand.
You understand? It's okay or not? So
Yeah, it's okay.
So the idea is you put a random sequence and you collect only the sequence among this random sequence that are able to interact with the protein. So all the ones that are at the bottom does not contain the right sequence for the interaction. So if you purify this band, you sequence the DNA that is contained in this band, you should get only the random sequence that is able to be recognized by the transcription factor. We do several rounds. It's a technical issue. It's only to be sure that the purification is almost perfect. But at the end, it's like if you have your transcription factor and you propose to the transcription factor different DNA sequence. The transcription factor can recognize a single one. When you recognize this single sequence, it makes the complex, and you purify the complex just by cutting the gel, and you directly sequence the DNA. So from that, among plenty of different sequences inserted randomly in this probe, you will purify only the sequence that are able to be recognized by the transcription factor. And so this is why it's called binding site selection. You understand the idea. So you propose random binding site and you select only the binding site that are recognized by the transcription factor. And at the end, you can have, so you do that plenty of time, you sequence plenty of different fragments, and you will get this type of analysis. Did you ever see this type of representation with nucleotide like that? Okay. So once you clone all the fragments, at the end, you don't have a perfect consensus sequence. You have the sequence preferences of the transcription factor. So it's usually represented like that, and it shows you the percentage of occurrence of single base at the binding site. So it is like that. For example, so it's coming from that. We made several rounds of purification, and at the end we have this consensus sequence. And how can we analyze this? It means that your transcription factor is able to recognize this is the consensus sequence of the recognition site. And you have to read it in this way. It means that the first position So I tell you randomly, it's in, let's say 60% of the case, it's an A, 30% of the case is a C, and sometimes it's a D. Second position, the same. For example, the fourth position of this site is mainly all the time a G. There, not the last one, but before the last one, it's almost all the time a C, and the last position is almost all the time a A. So you have the consensus sequence of the binding site recognized by your transcription factor. You understand the strategy? So in that case, it's very interesting because you start from the transcription factor. So this is what I was telling you. And you want to know which are the target genes. So from that, you do this binding site selection. So you manage to have the consensus recognition site of your transcription factor. And the next steps would be which genes contain in their promoter such a sequence. And from that, you can expect that all the genes that contain this regulatory element in their promoter region may be under the control of the transcription factor that you use at the beginning. But you have to take this result with extreme care. You see this sequence. If you count the base, it's 2, 4, 6, it's 8 base, okay? So you have to keep in mind that finding 8 base sequence on a genome, you will find it almost everywhere, okay? This is the problem. So when you want to search for such motifs inside the whole genome sequence, you have to take a lot of care in your searches, because you are looking for this sequence only, first thing, only in promoter region, because it's supposed to be a regulatory element, and so on and so on. So it's quite difficult to find only using this consensus sequence all the genes that are under the control of this transcription factor. But it works very, very well. Okay, nevertheless, at the end, when you did this kind of approach, which is finding the binding site of your transcription factor, in all the cases, you need to confirm this finding. And to validate this finding, you need to do it in vivo, okay? To be sure that your transcription factor is really able to recognize this sequence. And this can be done using, and here we see again the yeast-1 hybrid system. You can use it as a validation tool. Do you understand how can you use it as a validation tool? You understand? It's okay? So,
Yes.
for example, you have one transcription factor, you do binding site selection, and your conclusion, it is this consensus sequence. After, to validate this finding with yeast-1 hybrid system, what you have to do is to take this consensus sequence, the one that you found, you clone it in tandem in the reporter plasmid of the yeast-1 hybrid system, okay? The transcription factor that you use to find this consensus, you clone it in the effector plasmid of the yeast-1 hybrid system. And if they really recognized in vivo, so your transcription factor binds to this sequence, you will have the transcription of the reporter gene inside the yeast, and the yeast will grow on a minimal medium. You understand the idea of validation? It's okay?
Yes.
Okay. But I don't want, but think about one thing is we are starting back using one hybrid system with all the pros and the cons. So you have to take care also. The other way to validate the the binding and the validation of the interaction of one transcription factor to one cis regulatory element, and to prove its functionality is to do what we call the transient co-expression in the homologous system. And for that, I took back, so I don't know if you remember, maybe, okay, I will change the PowerPoint document. Just want to, okay. I just changed the PowerPoint. Okay. Do you remember when we look at this case study, when we did the analysis of the promoter of this gene that was specifically expressed in the vascular tissue of the plant? So we did the promoter analysis. Okay, and at the end we found this regulatory element that was necessary and sufficient for driving the activity. You remember that? Then from that, we did all the EMSA experiment, and I showed you that there was protein DNA complex that was formed in this regulatory element. Okay, perfect. So this is a little bit the same thing that we saw in the exercise with the growth hormone and the promoter. So from that, there are no slides on that, but the cDNA coding from one of the transcription factor has been cloned. Okay, so at that steps, we know the target gene was this CCR gene, okay, the gene that in its promoter contained this motif. So we know the target gene, and we know the regulatory region of this gene, which is just written down there. And we know that in this region, there are proteins that are able to bind to this region. And the cDNA of this transcription factor have been cloned and sequenced. And the next step was to do in vivo transactivation assay. So the idea there was, okay, now we know the target gene, CCR, so that contained the binding site. So if you look at the position, this is exactly what we saw for the analysis. So basically, this gene, which is the target gene, all the data before showed that the cis element were this small 50 base pair region here. So this is the reporter construct. What do we do for this reporter construct? You clone the promoter of the target gene in front of a reporter gene. Okay, from that, there is no problem. So we are now in a situation where if we, in transgenic plant or in transient expression, because it's a plant gene, if we bring the transcription factor and if we overexpress it, we should activate the transcription of the reporter gene. Okay, so if we overexpress the transcription factor, we should overexpress the reporter gene. So the reporter construct are done to monitor the reporter activity, and then we have effector construct. So effector construct, we are going just to look at the first lane here, what it is. So from CRMV35S, it's a constitutive promoter. Okay, so we have a promoter that is going to transcribe constitutively the transcription factor. And the transcription factor, it's R2, R3 plus C-terminus. So this is the full-length transcription factor. You take cells, and so plant cells, plant protoplast to be precise, and if you transfect the reporter construct plus the effector construct, you will be able to monitor if the activity of the transcription factor is able to activate transcription or not. You understand the strategy? So it's a little bit like in the yeast-1 hybrid system, but with completely normal sequences. So you have a reporter construct that contains the target promoter. You've got an effector construct that overexpressed constitutively the transcription factor, and you transfect the cell with these two constructs, and you check for the reporter gene activity. So we are going to compare, well, it's not a problem, we can compare the gray bars in this graph. So there is the control. The control, it's written PGR1, so it's the name of the plasmid, it's not a problem. And the positive test, which is when we overexpress the transcription factor. In that case, the transcription factor, it's a MIB type transcription factor. It's called MIB2. Okay. The control, what it is? It's an effector, so an effector plasmid, so a constitutive promoter, but no coding sequence after. So in the control, we only introduce the reporter, but the effector does not produce any transcription factor. In the EGMIB2 plus test, we overexpress the full-length transcription factor inside the same cell where there is the reporter gene present, sorry. And what you see is that you start without any transcription factor. So this is a relative activity. It's the unit is percentage, so it's not a standard value. So we have to consider the negative control, which is at 100%, as the reference, which means no activity, because we don't overexpress the transcription factor in that situation. And if we overexpress with the construct, with the reporter construct, if we overexpress the full-length transcription factor, in that case, we see that from the basal activity of the reporter gene, we increased by 2.5-fold the transcriptional activity of the reporter gene. So that type of result will be, okay, when we overexpress this transcription factor, it can recognize the cis regulatory element present in the reporter construct, and the consequence of this recognition would be the transactivation of the reporter gene. So from that type of experiment, you can say two things. Your transcription factor, in that case, this MIB2, is able to recognize this reporter promoter. So this is a good point because we are looking for this. And second result is when this transcription factor recognizes this promoter, it can activate transcription. Okay, so this is the final proof of the transcriptional capacity of transcription factor to activate transcription. Is it okay for you, this? Yes or no?
I think yes.
Yes, but I want you, we're going to finish with this. I want you that we think about, okay, I'm really sorry to have to change of slides all the time, but okay, I don't know what I am sharing now. No, it's not that one. Sorry, I need to close some windows because, okay. Really sorry. This one, okay. So I just want that I'm going to explain you one thing just for the last two sentences in this. So you have, let's make a summarize. You have a transcription factor. You can find out which site is going to recognize, and using the one hybrid system or what we see the co-expression, we can see if this transcription factor on the top of recognizing that sequence can activate yes or no the transcription. So what I want to discuss with you is I wrote down two strategies, the one that is based on yeast one hybrid system and the one that is based on the co-expression in homologous system. It means in animals or in plants if you are working on that. So according to you, what is the main problem and the main differences in using these two strategies if you want to really validate an interaction and the consequence in transactivation? What would be the strategy and the problems? Do you have an idea? I'm going to share the whiteboard. You don't have any idea? It's exactly the same problem that exists between when you want to isolate the cDNA of a transcription factor. The same problem that are the difference between purification and one hybrid system. So,
This is for the heteromer.
Exactly. Yes, exactly. But not. It's the same problem, but the origin is a little bit different. So let's say that you have a transcription factor. Okay, perfect. So I make a say. This is in the yeast. Okay, the yeast one hybrid. Okay, you know the transcription factor. You know the target. Okay, and you will see if the transcription factor, when it recognizes the target, it can activate the reporter gene. Okay, if it works, if this works, it's okay, perfect. You have proven that your transcription factor can really recognize this sequence, and when they are bound together, it can activate transcription alone. So as you said, the problem would be the heterodimer, because if we need to have heterodimer, it's very much more complicated because we can have only one cDNA in one yeast. You understand the problem? If we are using co-expression experiment in homologous system, what is going to happen? So, co-expression. For co-expression, it's more difficult to set up, but it's more powerful because what do we have? We have the cell, okay? You transform the cell with the reporter construct. So this is the place where your element, your cis element here is, okay? You have, so I will write it in blue. You have another construct that is going to express your transcription factor. Okay, so this is the effector. So from that, you have the protein, which is the transcription factor. Okay. What can happen is that you are trying to prove that this transcription factor can recognize the sequence, and then once the recognition took place, it can start the transcription of the reporter gene. Okay, so this is the point. But imagine the situation in which your transcription factor, the one that you are working on, need to be in interaction with other transcription factor or other protein that are normally present inside the cell. Will it work in the co-expression experiment? So keep in mind that this is the cell from plant if you are working in plant, from animals if you are working in animals. So this is homologous system. It's very important because since you are in the system in which the biological system in which the full system is working. It means that inside these cells, you have all the other components in terms of protein that are necessary for creating the complex. Because, for example, in the cell, you have all the other protein already present, and to be active, your transcription factor need to have all the three partners. And in that situation, you don't have any problem because the effector plasmid that you transfect in the cell, it's here to overexpress your transcription factor. But in the cell, naturally, you've got all the other protein partners. So the complex, the heteromeric complex can be formed inside the cell, and then the activation of the transcription can start. So you see, when expressing a single partner, because the other partner can be present in the cell, you can reconstruct the complex and then start activation. And this is not feasible if you use yeast-1 hybrid system. Okay? You understand? Because in yeast-1 hybrid system, you can only bring inside the cell one of the protein partner of the complex. So the full complex heteromeric will never be reconstructed again inside the yeast cell. Do you understand the differences between the two? It's okay? So always the same. Yeast-1 hybrid, yeast-1H, 1 hybrid system. It's very good. It's not a criticize. It's faster, but you have some situation in which it will never work. Whereas the co-expression is a little bit more complex to set up. You have same construct to clone, but you need to introduce to introduce all this in the cells, then to monitor the activity. It's a little bit more tricky to set up experimentally. But at the end, you take advantage also of being in an homologous system, which means that all the other putative partners are already present in the system. It's okay? So it's always the same. You have different strategy and you have to make a choice in which one to use. Do you have any question on that? Because we are going to finish. I want just that we look at the last slide and we will have finished for this part, for the confirmation of activity. Do you have any question for that? It's okay. So just to finish, because I think it's important that you have that in mind. So I will explain you quite quickly what is drawn in this slide. But before, I want to explain you something. So what we are trying to do is, once we have isolated a transcription factor, the first thing that we want to do, and this is what we are trying to do for a moment now, is to validate its activity. Okay. But there is very, very big problem with transcription factor, because you understand that one transcription factor can activate plenty of different target genes. So even though you are working on one specific target gene, you can have, if you overexpress one transcription factor, you can have very damaging side effect because you are activating other genes. Okay? The second point that is very complicated with transcription factor is sometimes if you overexpress a transcription factor at the right place in the organism or at the wrong time in the development, you will kill the plant or the animal, because there are some transcriptional activation that doesn't have to be active at certain steps. Okay, so it's always very dangerous to constitutively overexpress a transcription factor in a wild type system, because there are a lot of damages that can occur. So in order to avoid constitutive expression, there are researchers that created an inducible system, and this is very good, this inducible system, because it's a very powerful tool. Actually, it's just to show you how it works. And you can use this system in order to overexpress whatever protein you want, but it's very useful for transcription factor, when you want and at the time you want and at the place you want. So it's an inducible system that is based on the glucocorticoid hormone and glucocorticoid receptor. So did you ever study this glucocorticoid receptor? No? Maybe not?
Not yet.
Okay, anyway, what I want to show you, so you don't need to remember all the names there, but I want to show you the strategy. So, for example, we want to have a system in which we have the total, we can turn on or turn off artificially the expression of the gene of interest. And it's very powerful because we can directly choose when and where we want to have the transgene that is going to be expressed. And this is done using this natural system that is present in animals, which is the glucocorticoid receptor. It's called GR, okay? And an artificial corticoid, which is called dexamethasone. It's written, I don't know where. Okay, well, it's Dex. Oh, sorry, it has jumped. And the system is working like that. Okay, so if you don't treat the tissue with the hormone, there is no activation of the gene. If you add the chemical compound, the glucocorticoid, you will activate transcription. You see here it's written not expressed and expressed. And you can, this is the target gene, so this is the gene that you want to overexpress. It can be any kind of transcription factor, no problem at all. So what is done is this kind of system. Just keep in mind the concept. Don't remember HSP90, LEC2, and so on. It's not a problem at all. So the hormone, dexamethasone, here is the little yellow spot there. And the receptor of dexamethasone is the GR gray ball here. And in this system, it's a natural system in mammals. The receptor is in the cytoplasm. And the receptor is able to migrate from the cytoplasm inside the nucleus if it is bound to the hormone. And once inside the nucleus, it can activate, because it behaves like a transcription factor, it can activate the transcription of the target gene. Okay? So what is done is like that. You have a system where you make a construct in which you will be able to overexpress your transcription factor, but only when you want and where you want in the plant or in the animal tissue. So you transform the organism you are working on with this kind of construct. It contains this construct as well. And once you will add dexamethasone to the cells that you want to overexpress the thing, the glucocorticoid receptor will recognize and bind to the dexamethasone. The binding of this dexamethasone will remove one partner of the complex which is in the cytoplasm, and the removal of this partner, HSP80, is going to have as a consequence the nuclear import of the complex. And when this complex glucocorticoid receptor LEC2 is inside the nucleus, you're going to activate the transcription of the target gene. So I put this slide only to show you that we have, and those systems, they are working in animals and in plants very easily. So this is inducible system that are very often used to test the activity of some protein, so the protein that you are going to express, but it's very useful when this type of protein can be lethal at certain stage of development. Okay, so and to finish, I will show you one advantage of this inducible system when we want to study the targets of transcription factor. So I show you something. So we can say we are in two situations. The first situation, so we don't put the inducer, we don't put DEX, okay? And I will summarize it like that. You have your transcription factor, which is under the control of DEX. Okay, so I will put GR like that. Okay, so first situation, okay, you have the living organism in which you are working. It contains this type of construction, so a transcription factor under the control of this inducible promoter. So it means that if you add the dexamethasone, the transcription factor will be transcribed and translated. So if you don't put, you don't have anything. Second, so the idea is to find the target genes of this transcription factor. Okay. Second step, you do a test where you add the hormone. What is going to happen is you're going to have transcriptional activity and you will produce the protein of your transcription factor. This transcription factor is going to be able to activate different genes, which are the target genes. So gene 1, gene 2, gene 3. Okay, let's imagine that the same transcription factor can activate 3 genes. Okay, so you are in a situation where you can control the transcription and the translation of your effector, the transcription factor, and this transcription factor is able to activate a different set of genes. So what can be done if you want to know if the question is what are the targets of your transcription factor? What you can do to answer to this question is to compare the genes that are expressed when you don't treat with dexamethasone to the situation of the gene in the same cells that are expressed when you are adding dexamethasone. So the difference, the genes that will be differentially expressed in these two situations will be only the genes that are under the control of this transcription factor. Do you understand this idea? Because we have an inducible system. So you have the tissue or the cell at a specific state. You induce the production of the transcription factor and you look for which are the new genes that are going to be overexpressed. And from that, all the new genes that are going to be overexpressed are going to be putative targets of the transcription factor. Is it okay? Yes or no? But there is a problem, a small problem, and we finish with this. Okay. Do you understand that a situation, no induction, so you don't induce these genes, you add the hormone, you induce the target. So you can say that G1, G2, and G3 are targets. But you have to be extremely careful with this because they can be primary targets or indirect targets. I explain you one situation. You have your effector construct that is able to overexpress your transcription factor using DEX. Okay, so it's a summary. So if you add the hormone through the GR receptor, you activate the transcription factor. Okay, and you have the protein, the transcription factor. This transcription factor can activate, as I was just drawing before, different primary targets, genes. Alors, what I call primary targets are genes. So gene, I draw everything and I explain. Gene number 1 and gene number 2. And I will say this is primary target. So you induce the transcription translation of the transcription factor. This transcription factor can recognize specifically the regulatory element of gene 1 and of gene 2, which are primary targets of the transcription factor. Perfect. But what can happen is that gene number 2 also code for another transcription factor. I would call it transcription factor number 2. And this transcription factor, as you may expect, can activate other genes. So gene 3 and gene 4. Because this transcription factor can bind to new regulatory sequences present on G2 and G4, G3 and G4. Is it okay? Do we understand this drawing? So
Yes.
these G3 and G4 are secondary target. Okay, so it's perfect. The situation is perfect, but we can control the production of the first transcription factor. This first transcription factor can activate the primary target directly, okay? And one of the primary targets can also be some transcription factor that are going to activate secondary target. And what you want for your research is only to identify the primary target. How would you do that? And we will finish the lecture today only with this. Do you have an idea? I give you a clue on that. This is using drugs, specific drugs. Do you have an idea? So, okay. I explain. Tell me.
It will be a lot of work. But,
yes. No, but one private thing. To be clever is also to be lazy, because a lot of work, I agree, but there are shortcuts. Okay, so let's understand it again. When I put the DEX here, you activate the transcription. Do you agree? Yes or no?
yes.
And the translation. So you activate everything. Okay, so the transcription factor is there. It's going to activate the target genes, primary targets. Among the primary target genes, okay, you have one transcription factor that can activate the secondary target. So what is happening is, if we manage to block step here, okay, we will have only the primary and not the secondary. You understand?
Yes. How?
How? So we have a drug, a very powerful drug, which is called, so you find it CHX. Did you ever see that? It's called cycloheximide. Did you ever see?
No.
So it is an inhibitor of translation. Translation, not transcription. So what is done is you treat with DEX, okay? The transcription factor is transcribed and translated. Very important, it needs to be translated, okay? So it is translated, but so you leave, so the idea is you treat with DEX to activate, to produce the first transcription factor. A few minutes after, because it's very, very fast, transcription and translation, and to be sincere, it's something like between 10 and 15 minutes. So just after adding the DEX, you treat with cycloheximide. Cycloheximide will block all the translational process in the cell. So it means that only the protein that are already present can be active. So because cycloheximide has been applied after the translation of the first transcription factor, this transcription factor can be active to activate the primary target. So there will be transcription of the primary target. So this is how you identify them. But because of the cycloheximide, the mRNA of the G1 and G2 will never be translated. In other words, the transcription factor number 2 cannot be translated. So you will never activate the secondary targets. Do you understand?
Yes.
Okay, so if you understood, so it's, you see, conceptually, it's very simple but the result that we have from that are very important because it's the only way to make the difference between the primary target of your transcription factor from the secondary target that can come from this situation, for example. Okay, because in the idea of you are working on this transcription factor and the result that you want are only the primary target and not the secondary. So what I would like, what I wanted to show you is this, is using a drug-based approach, we can separate the primary target of a transcription factor from the secondary target in the transactivation process. Do you understand the thing? So it's a way to do this kind of analysis. Is it okay? And do you have any question? So we saw a lot of techniques today. Do you have any question before that we finish? Because it's 6 p.m. for you. Yeah, it's 6. No?
I don't really question. Do you have any plans for exams or anything?
What do you mean plans?
Maybe like timing, schedule.
The schedule. No, I don't know yet the schedule because I need to see with Hang which are the slots that are possible because you have other lecture in parallel. So the date is not planned yet, but it should be done. I can tell it to you before Friday. Anyway, when do you have the other exams for the other teaching units?
Right now, we only have one for tomorrow morning. And
So you have exam tomorrow morning.
yes.
Okay. Wait, because I've got all your timetable in my laptop. So yes, tomorrow morning you have exam with 1. Is that okay? Is it
Yes.
this one?
Yes.
Okay, for introduction to pharmacology. Okay, and then the next exam that you have, and the coming week, you have only on Friday, BP 13, it's the exam on Friday, next Friday morning for you. Is it okay?
Yes, I think so.
BP 13, it's, I have it on the timetable. It's 11 a.m. on next Friday, on the 28th. You have final exam BP 13.
Yes.
Yes, it's okay. And then the week after, you don't have any more lecture. So I would say, okay, I was at the timetable. I would say the exam. When are you going to start the internships? Okay. No.
In
Okay.
April, I think.
Yeah, it's okay. So I would say that the exam for BP10, okay, I don't have a precise date, but we can plan it if it's okay for you in the first week of February. Is it okay?
Oh, it's Tet holiday in Vietnam.
Ah, yeah, it's that. Okay. Because this year the TET is on the 1st.
Yes, the
Okay, first. Back on Monday the 7th. Is it right?
Yes.
Okay. So, well, it will be after that, and I will try to find a date in the week where you don't have a lot of lecture. Okay. So let's say starting from the 15th of February, is it okay for you, or do you want to have it later on?
That far then as later. I'm just concerned whether it's going to be like really, really close and we have to be prepared for
No,
it.
okay, I agree, but listen to me. When will it be for you the more convenient to have the exam? Because I don't have any reason to put you the exam at a moment where you have plenty of other work to do. It's better that you have the time to study, and this is very important. So you would prefer to have the exam in March, something like that, or before?
No,
Before.
before.
So second half of February or first half of February?
First half. Because we
Okay.
will have
Okay.
lecture.
Okay, so listen, what I'm going to see with Hank is try to find a date in the first half of February, and we will try to find a date that is convenient for you, not to have a lot of things to do at the same time. Okay? Is that okay like that?
Yes.
Okay, so let's do like this, and I will tell you before Friday. So tomorrow we start at 3 p.m. for you, but we will have only 2 hours of lecture, so we will finish 1 hour before because I've got another meeting just after. Tomorrow we are going to finish the part of the exercise, and we're going to see different methods for analyzing gene expression. So we're going to do qPCR, RNA-seq also for genome-wide analysis. And on Friday, we will finish the lecture with everything that is related to post-transcriptional gene silencing. And this is where we're going to finish the model of the central dogma. And I will show you a big drawing when we put together all the things. And if you have time before Friday, on Friday, I will keep, let's say, almost 1 hour. If you want that we come back on specific point of all the lecture that we have all together. So if you have question, I will take time. We can discuss. If you want me to explain you more in detail some points, we can see that on Friday at the end of the lecture. Is it okay? Like that?
Yes.
Okay, perfect. So see you on tomorrow afternoon, so 3 p.m. tomorrow. Okay.